just so I'm clear again, so you're saying it was a point mutation in one nucleotide, but that caused the snipping out of, what did you say, about 40 or 50 amino acids or something in the protein? Did I get that right?
Yeah, well, there is a specific point mutation in the lamin A gene occurs, and that leads to an alternate splicing event. It sets up a different splice. The splicing mechanism recognizes it, and it jumps ahead to the next exon, but it leaves out a region of the gene that it includes 150 base pairs or 50 amino acids in this region. And within the region, the normal lamin A, when it's processed, gets a farnesyl group, a lipid group, added to the end of the polypeptide, and that gets it into the nucleus, and then normally that gets removed by a splicing of it, by a clipping, removal of the terminal region of this polypeptide. That doesn't occur in progeria because it's missing this internal cleavage site, and that has the effect of leaving the farnesyl lipid attached to the end of the polypeptide of the protein, and that causes it to gum up the works. So
when you say gum up the works, you mean it continues to accumulate?
Yeah. Well, it's a pathological protein at that point. It's not working normally, and it has the effect of lamin A is a very important protein that coats the internuclear membrane of many cells in the body, most cells. It doesn't for the brain, but it does for most of the body cells. And the progeria patients actually have pretty normal mental abilities and actually have bright little kids. So it doesn't affect the brain for an alternative reason, because they have a microRNA that's involved with it in the brain. But in the body, it leaves this farnesyl group on, and that causes the lamin A to not function normally as it's supposed to. It acts as it attracts chromatin, and it brings a lot of factors in the nucleus together, and that's not working normally, and it causes the cells to show nuclear blebs, kind of a pathological feature you can see in the microscope, and it interferes with a lot of metabolic functions in the body, and that's how they develop. They're dwarfed, and they have heart disease that develops when they're 7 or 8 years of age, and usually die of a heart attack or stroke with an average age of about 14. And they're dwarfed. They weigh, no more than about 30 to 35 pounds, or 60 or 70 kilograms, even when they're teenagers. So it's just a molecular mechanism that was, discovered, and this is the mutation. And lamin A actually has about 10 other different mutations that cause different diseases, and it's quite remarkable in that regard. Lipodystrophy, a certain form of muscular dystrophy, and various other syndromes that are not progeria, but are kind of in the family of a problem, a genetic problem.
So does this mean that it's a good candidate for gene therapy, some sort of intervention?
Yeah, they're
some CRISPR thing or something.
Right. They've actually done CRISPR in the mouse and cured it. And there was a nice paper, I think, in the New England Journal or Nature in January showing it could be done. The problem is that CRISPR still causes off-target effects, and it can cause cancer and things that they haven't yet, approved it for human use. There are morpholinos that are kind of synthetic interfering RNAs that companies are working on that look quite promising as a drug that could be given. And there are several other strategies that are in the works.
Well, I suppose it's a problem in terms of producing drugs because it's such a rare thing that there's not much money in it.
Well, Well, I mean, again, it's kind of the model for heart disease, so there's a big interest in it from the cardiac standpoint as well, so that if you understood how it could be treated and how it's affecting cardiac function and all, and then it might have more general use. At least that's the idea. Yeah, it's it's a fascinating disease, and it's very striking to see a child with this.
Yeah, I've seen the photos.
Yeah.
Can I ask a question too? And it might, perhaps because I don't understand some of the biology that you mentioned before, some of the more detail. But if you were to take a person with progeria and you were to take them at, say, when the age is theoretically, maybe they're 5, 6, 7 years old, I don't know what it is, say they're equivalent to a 60-year-old normal person, is everything roughly the same? I mean, are you really talking a disease that is talking about rapid aging, or are you talking about something that really just gives the symptoms or looks like there's rapid aging? That's the piece I'm not completely clear on. Like if you compare with a 7-year-old progeria patient with a 60-year-old normal person and then a 12-year-old, et cetera, do they seem to be the same as that age?
Well, I mean, it's not aging; it's a disease, and
yes,
it's distinct, but it has a lot of features that resemble aging. I mean, they have
alopecia. I understand.
They lose their hair, and their skin looks quite aged, and
so it's a disease that resembles aging rather than a rapid aging in itself. Let me put it that way. I'm not technical enough to understand, but that's why I'm asking in very simple terms. Thank you.
Yeah, it's a model for aging, a human genetic model for aging, but it's not aging. It's a disease, and that's it's distinct. But it's.
So you can't say that human aging is the same thing, but happening at a slower rate. I mean.
You can't. I mean, you can't.
I understand. Yeah. That's what I just needed to clear in my mind. Thanks. Sorry.
My experiments here in Sydney were to try to establish that there is actually a connection with progerin building up in mice and normal age, and then in normal aging mice, and seeing what could be done about aging in mice, putting them on a special diet that promotes longevity. And but we didn't find it, and the reading I've done suggests that, we're not going to find it in the blood like we were looking for, or even in looking at liver or different tissues that we looked at, but it might be present at low levels in a certain fraction of fat cells. That's what Maria Eriksson, who's an investigator in Sweden,
understand.
Has been finding that, with her technique of RNA sequencing or something, that she can show that within a certain population of fat cells, that they show a higher level of progerin in normal human aging. But it's kind of limited in its applicability to aging.
I see.
But, it's still fascinating, and it is, once the mutation was found, then right away there was a potential treatment that was available. This farnesyl group gets added by a special enzyme, and there's a way of blocking that enzyme. And it's a drug that was being used. Certain other proteins get farnesylated as well, including a family called RAS, and they're involved with neural tumor, with brain tumors in children. And they were doing a trial of Larnafarnib, which is this drug that inhibits farnesyl transferase, which is the enzyme that adds the farnesyl group. And so it's been given to the progeria patients. The foundation has sponsored a large-scale study, and it improves their survival, by 80% for a limited period of time. But that's not a cure. It's so they've added a second drug, a rapalog, rapamycin. It's everolimus, it's been found that rapamycin increases the longevity of mice when it's given. I mean, longevity in mice, the basic aging discovery a long time ago was that if you put them on a calorically restricted diet, they live longer. And then they found you could do the same thing with putting them on rapamycin. Rapamycin is a drug that's been around, and it's been used for humans that have had organ transplants to inhibit their immune system. it inhibits the mechanistic target or mammalian target of rapamycin. mTOR is the main, and mTOR is inhibited by rapamycin, and mTOR is a basic like enzyme system that's, if you have too much of it, you grow more and you have a higher incidence of cancer. If you inhibit it, you go into kind of a survival mode and you live longer, but you might have, immune problems if you take too much of it. And some of the people are doing trials of it to see if you can take a microdose and see if that helps with longevity in humans.
One question I've got for you, Ted, about the cryonic side of it is that you got into this whole scientific area, medical area, because of your interest in being frozen. How is your personal view about freezing tissues and reviving them at some stage in the future? Has that changed at all over the course of your career?
Well, I mean, when I was doing research in the blood lab at Mass General, and then thinking about it, it seemed to me that it's just a technical problem of figuring out how to freeze an organ and bring it back, and that if we could freeze kidneys and hearts and use them for transplants, it'd be and I just figured eventually this is just a mechanism that needs to can be discovered in some way that you could do that. But it seems when you freeze an organ and then try to reperfuse it, there's a lot of endothelial swelling, the blood vessels swell and they prevent circulation. So, it just seemed that, so I just kind of lost interest in it, thinking that even, if I was brought back, I'd still be aging, and what would I do about the aging problem?
Oh, you've got to reverse the aging as well.
Yeah. And so I got more interested in that, but, it's still, it'd still be great to be able to freeze a kidney or a heart for transplant.
I don't know if you've seen it, but there's been a couple of papers over the last few years about. I read Fay's paper about trying to freeze a kidney. Yeah, and it sounds and they've done a large mammal kidney as well, I think. I think they did a sheep kidney or something. Or something.
It hasn't yet gotten to the point where it's become, standard of care.
No.
In the medical field. So,